Alrighty, welcome to chapter 26, which is the urinary system. The kidneys are one of the most interesting organs in the human body, in my personal opinion. They don't get as much credit as they deserve. There's two for a reason, because one does quite a bit on its own already, and having two is just kind of impressive. Once we start talking about how much work the kidneys actually do for us, I hope you'll be just as impressed as I am. I really enjoy this cartoon from the Awkward Yeti, so I hope you do too. As per usual, we have our purple slides, and we're going to start with the anatomy. So for this chapter, we nicely go from a pretty large view of what the kidneys are doing, and then down to a more molecular view of what the kidneys are doing. So we're going to go from the outside in. Let's start with the kidneys. So the kidneys are paired. They're retroperitoneal, as we've discussed in chapter 24. One is a little higher than the other. The right one is going to be a little lower because of the liver. That'll push it down a little bit, which changes the vasculature in that area just a little bit. And then the left one is going to be a little higher, but usually they're about the same size normally in a healthy person. The kidneys are what's going to be producing urine, and urine is essentially water, ions, and some small soluble compounds, and we'll go more into detail later. And then once the urine is made, it goes through the urinary tract, which is a paired set of ureters. Then it gets stored in the urinary bladder, and then whenever we're ready to consciously release it, then we release it through the urethra. Urination is also called micturition. So functions of the urinary system: excretion, so excretion of ions and water that we don't need, elimination, excretion from the bloodstream, elimination actually out of the body, and then homeostatic regulation. So this is where we're regulating blood volume and blood pressure with EPO. EPO is released whenever there's a low amount of red blood cells. And then the kidney says, we need to make more red blood cells, so it releases EPO, and EPO goes into the skeletal system, as we've discussed in the endocrine chapter. The kidneys are also responsible for releasing renin, and renin is part of the RAS system, or the renin-aldosterone angiotensin system, and we'll talk about where renin is specifically released from in this chapter. And as we know, renin goes and converts angiotensinogen into angiotensin 1. The urinary system also is tasked with regulation of blood plasma for sodium, potassium, chloride, and other ions, but those are the main three that we really care about and that we'll talk about in this chapter. It also pays attention to calcium levels as well. So we'll talk about how things get reabsorbed or secreted, and please don't let this be confusing. If we are secreting something, that is going into the nephron, which we'll talk about, so that's going into the pre-urine. And if we're reabsorbing something, then that means we're pulling it back out of that pre-urine and putting it back into the bloodstream. All right, so other functions, we're stabilizing blood pH as well, because we'll be moving some hydrogen protons and some bicarbonate ions. We will exchange some valuable nutrients for things like urea and uric acid. These are different in each species, so this is why like birds' poop is more white and solid consistency, because they have different not composition but percentage, what's the word that I'm thinking of, they just have a higher amount of urea and uric acid in their urine than we do. There's also mixes with feces as well, so that has something to do with it too, but these are components that are relatively toxic to humans, and we don't want to keep any of those around, so we'll eliminate those. The kidney is also responsible for assisting the liver by detoxifying poison. So whatever the liver can't really clear out of the blood, it's really up to the kidneys to do. The liver is more concerned with a wider range of nutrients and ions, while as the kidneys are pretty specific to the ions and nutrients that it's collecting. But if it helps you remember, the kidney is more focused on, I don't want to say more important, but generally more important ions to collect rather than the liver. So in a way, it's a little bit more important, I should say. Also think about relatively where the kidneys are. So the kidneys have their renal veins and arteries. Those are coming right off the abdominal aorta or the inferior vena cava. And these are retroperitoneal, so they have nothing to do with the digestive system. So the liver is going to clean out everything, or clean out the blood that interacted with the digestive system, versus the kidneys are going to clean out the blood that came from everywhere else. So the kidneys kind of have a bigger volume to deal with at any given time. This view on the left is a posterior view, where you can see the left one is again a little higher than the right one, and then a transverse section on the right-hand side to see where it is in relation to the spleen or the pancreas. They're about in the same plane. The kidneys have a protective tissue covering of connective tissue. It's called a fibrous capsule. And then there's something called perinephritic fat. So nephritic is referring to the nephrons in the kidney. Peri meaning around the nephrons in the kidney. This is kind of not the best name for it. I'll show you a model of where we can actually see that. I don't know what else I would name it, but it's but it's a little fat cushion within the kidney. From the cadavers that I've seen, I haven't seen this be any more or less fatty than depending on if the person was more fatty or not. So this seems to be a pretty consistent. Sorry about that. That was my dryer. This seems to be a pretty consistent layer of tissue. And then we have the renal fascia. This is just like all of the muscular fascia that you learned in 201. This is going to help protect and keep the kidneys in place in the posterior section of the abdomen. So here's an anterior view of the kidneys showing their blood vasculature supply. So coming right off of the inferior vena cava and the abdominal aorta, and how they both have their own set of paired renal arteries and veins. Here's looking at one kidney, on the left a cartoonistic depiction, and on the right a cadaver depiction, where you can see the renal cortex, cortex meaning bark, so it's going to be surrounding the kidney. And then on the inside we have these little triangular pieces. Those are called renal medulla, singular, medullas, plural. The inferior part of the renal medulla, or the bottom part of the triangle, is called a renal pyramid. So you can imagine that urine is going to be made in the top part of the medulla and then drain into the pyramid, and then from the pyramid it'll drain into the minor calyxes, and then from the minor calyxes, or calyx singular, it'll dump into the major calyx or calyxes, and then collectively that will all dump into the renal pelvis, and the renal pelvis will then dump into the ureter on both sides. We also have a hilum here, so remember to be specific whenever you label hilum in the lab practical. It's not just hilum, it's hilum of the kidney. And the hilum is just identifying the collective vessels or ducts or tubes that are going in and out of the kidney. So the hilum would also include the artery and vein. That's just not pictured here. So artery, vein, and renal pelvis would be part of that. Let's go ahead and jump into complete anatomy and see what these look like as well. This picture is going to be of the abdominal. Oh, all right. Restore. Okay, maybe it doesn't want to restore. Okay, just 1 second. We'll go abdomen, and then we will add the urogenital system. Okay, perfect. So here is the abdominal region of the skeletal system with the two kidneys. You can appreciate that one again is a little higher than the other. In this picture, not so much. I think in an actual cadaver, it's a lot more obvious the difference between the two. And then after each kidney, you can see that they dump into the ureters, and we have those bilaterally, and then those are going to dump into the bladder. These tiny things coming off, this is a picture of the male pelvis, so these are the ductus deferens. We'll talk about that whenever we get to the reproductive system. And then this middle ligament is called the median umbilical ligament, and the reason why it's pictured here is because the distal attachment point is the urinary bladder, but we don't talk about that in this class, so don't worry about that. But let's go ahead and zoom into the kidney from what we just talked about. So I'm looking at the left kidney here. Here is, they're calling it the cap, but we don't call it a cap. And let's go ahead and hide those so we can reveal what's looking on the inside. I really like this view because this is really what the kidneys do look like. That cadaver picture also does a good job, but it looks a little bit more shriveled and less differentiated. But you can really identify with the naked eye the difference between the cortex and the medulla. So here we have the cortex, which is surrounding all of the individual medulla. This is highlighting it as the renal pyramids, but the renal pyramids are actually the, let me draw this, are actually the bottom portions of all of these, versus the medulla is the entire triangular looking piece. So please don't get those two confused. And then the medulla will dump into the minor calyxes, and then from the minor calyxes into the major calyxes, and then from the major calyxes into the renal pelvis, and then in the renal pelvis down to the ureter. There's nothing else really special to see from this view on this model, but we have much better, more microscopic views that we'll get to in a bit. Now, let me get rid of those drawings. Perfect. Oh, maybe not. It's the only thing I don't like about this app is that I can't erase the lines that I made. And so hopefully that will come with an edit, but we're not there yet. One more. There we go. Okay. All right, so now let's take a look at the urinary bladder. So we're going inferiorly. We can see that the urinary bladder sits more anterior within the pelvic floor. This is the same for males and females. The urinary bladder we can divide left and right. Let's take a look at the inferior aspect of it though. So right here, this is where the urethra would be coming out from. This little hole that has nothing coming out of it. The reason why it's not identified here is because they're trying to separate this from the genital region, which is just shown in different models, so that's why it's not here, but it is part of the urinary system. I'm going to go ahead and remove the pelvic bones so that we get a nicer view of what's going on in the bladder. And if we take out one side, we can see the inside of the bladder. It looks pretty muscular. It's a muscular organ. It's a hollow organ. This is where all the urine is going to be stored. And it also has those folds that look really similar to what we saw in the stomach. These folds will also be important, but what I want to highlight is something called the trigone. It's called trigone because it's in the shape of a triangle, and at each of the top corners of the triangle is where the ureters will actually enter into and start to fill with urine. And then at the end of the trigone, the last corner of the triangle is where the urethra would be coming out from. All right, that's it that I want to show you here for a macroscopic view of the kidneys and the bladder. Let's go back to the lecture notes. Oh man, it's just not going as quickly as I would like it to. Sorry about that, team. Okay, all right. So what is actually going on within those medullas? So in the medulla, let's draw like a trapezoid-looking thing. Within the medulla, we're going to have functional units called nephrons. This is each individual nephron is responsible for filtering out blood and producing urine, but we have many, many nephrons. So all of these cartoonistic depictions are a little bit of a lie because we have way more than we can even depict in a cartoonistic depiction. And these aren't really relatively drawn to scale either. These are really, really tiny, tiny things. And they look all coiled and confusing already, but actually they're even more coiled and even more confusingly so. So we'll always draw it out in a more simpler view, but just keep in mind that these are literally layered right on top of each other, and there's a bunch of them. So in the, if we zoomed into the medulla, we could draw a really baby version of what the nephron would look like. So there's a ball, and from the ball we're going to have a tube that comes off of it, and the orientation of the tube is important because some of the tube goes straight, some of the tube goes down towards the pyramid of the medulla, and then it cuts back over straight again, and then it comes back up, and then it cuts back over straight again, and then it comes back down. So this is really important. We'll give more identifiers to what's actually going on in the nephron, but for now, let's just draw it like that. We have two different types of nephrons. One are cortical and the other are juxtamedullary. So cortical, you can imagine, are closer to the cortex. These are actually the majority of the nephrons that we have. And then the rest are juxtamedullary nephrons. So these are closer to the medulla. And so if I were to actually draw what this looked like, if I had a kidney and I had a renal medulla, and let's pick a different color. My cortical nephrons are going to be here versus my juxtamedullary nephrons. I'm sorry. The ones I drew were juxtamedullary nephrons. Sorry about that. Nephrons. And then the pink ones will be our cortical nephrons. So I'm just going to draw a squiggle. Okay. The kidneys at any time will receive 20 to 25% cardiac output. If you remember, compared to the brain is about 12% of cardiac output. And the kidneys are going to have hopefully a relatively consistent level of cardiac output, just like the brain would as well. It's really important that the kidneys get a lot of the cardiac output because their primary purpose is to get blood and filter it. So it makes sense that they would have more blood coming to or being allocated to those organs than most other organs. So 20 to 25 makes pretty good sense. All right, so how do these nephrons do what they do? They look small and a little bit strange, but they're actually super good at their job. They filter blood at relatively consistent rates, and these are all in different units for you, but 125 milliliters per minute. I think that's kind of hard to imagine what that looks like throughout the day, but 7.5 liters per hour, which is 180 liters per day. And this fish tank here is about a 100-gallon tank. I tried to find one that is like pretty representative of what you would see in like the dentist's office. And a 100-gallon tank is about 380 liters. So this is really what the kidney does every, day and a half, is filtering out that much blood. And that's just one kidney. So that means all those nephrons that we drew have to somehow magically do this consistently and manage a lot of volume of blood. And throughout the year, 65,700 liters, but I have no idea what that looks like visually. The fish tank just gives me a better idea. What rates do you need to know? You need to know the 125 milliliters per minute. Don't worry about the other three. Okay, so each kidney receives its own blood supply, and the blood supply is named nicely for where it's going, but it's going to take a little bit of practice. What I'm going to do is I'm going to draw on the right-hand side here, but I would recommend you take 3 sheets of paper, and we're going to draw 3 big pictures on each of them. So one of them is going to be the blood supply. The other one is going to be the actual structure of the ball thing that we drew up above. And then the last one is going to be actually detailing what's going on in the tube. So I would recommend you take out one of those sheets of paper so we can start with the blood supply. Okay. So let's start with the arterial supply first. And I'm going to draw a kidney over here first. Okay, so arterial supply. So we know that blood coming to it is coming from the abdominal aorta via the renal artery. From the renal artery, then we branch into a couple of what we call segmental arteries. So I'll just draw 3. Double A for arteries. From the segmental arteries, then we have to branch into each of those medullas. So what I'm going to draw now is like a zoomed in version of what those medullas would be like. So if we drew a medulla here, let's go ahead and zoom into what that looks like. So I'll draw it like this. And right here is medulla. And right here is the pyramid. Okay, so from those segmental arteries, you can envision that they start here at the bottom. And once they they change names once they start passing the sides of the medulla. We're going to call those interlobar arteries. From the interlobar arteries, then they're going to make a pass over the top of the medulla, and it's making kind of a curve, so similar to the arteries in the foot. There's another arcuate artery in the foot, but this is called arcuate artery. And then off of the arcuate artery, we're going to have some individual branches that go up to the cortex. So the first branch that I want to draw are the cortical radiate arteries. Oh yeah, okay, good. So these are the cortical radiate arteries. Okay, perfect. Just want to make sure we didn't miss anything else. Okay, so then the cortical radiate arteries, then we need to zoom into how those actually give off blood into the nephron so the nephron can filter it. So I'm going to draw a zoomed in picture again of if we were to zoom into a cortical nephron, for example. I'd have a little ball, a little stick, da-dum-dum, dum, like that. So we're going to zoom into there. Let's draw that out over here. So here we have our little ball, our little stick thing that we don't know what's going on with yet. And we have the cortical radiate arteries. Okay. Those are going to give blood to an afferent, A to arrive, afferent artery. And then the afferent artery will feed blood into something called a glomerulus, which is this capillary bed folding in on itself, dense network of arteries. Okay. And then that's it for the arterial supply. So, to review, we went renal artery to segmental artery, to interlobar arteries, to arcuate artery, to cortical radiate artery, to afferent artery, and then to glomerulus. So we went from really big to really small. Now let's go backwards and get the venous drainage from this. Oh, sorry, I need to mention one other thing. So from the glomerulus, then we're going to have and I'm going to draw this in purple because it technically has some oxygenated blood, some deoxygenated blood, but it's still called an artery, so I don't want to confuse you by drawing it blue because it's still an artery, but it's going away from the glomerulus, so we're going to call this the efferent arteriole. And the efferent arteriole is going to give off a big capillary network that will surround the entire nephron, something like this, and this is called peritubular capillary. Oop. So peri meaning surrounding, tubular meaning surrounding the tube, capillary because it does have oxygenated blood. Okay, from there now we're going to talk about the deoxygenated blood. And here we will draw an arrow kind of arbitrarily going to venules. And now we need to zoom back into our medulla over here, and we can draw venules. I don't know, here I'll draw tiny dots because they're pretty small. And then from the venules, we will go into, again, cortical radiate veins. Veins, double V for veins. And then from there, we will go back the back to the arcuate vein. And then from the arcuate vein, we again have the interlobar, but now vein. And from the interlobar vein, we don't have any segmental veins, so I'm just going to continue drawing these like this. So interlobar veins. And then from there, we finally dump into the renal vein. And from there into inferior vena cava vein. All right, so that's the blood supply that we'll need to know. What's really important to know, obviously the order of this, but what's really important to know are the afferent and efferent arterioles, and the glomerulus and the peritubular capillary. Those will come back up again later. In the PowerPoint slide, I have it written out. And in the bottom right-hand corner, you have a flow chart of the renal circulation from renal artery all the way back to renal vein. All right, this includes blood leaving. Last is nervous innervation, so how do we actually stimulate the kidney to do what it needs to do? It's going to get sympathetics, and it's going to get it from the celiac plexus. We didn't talk about the celiac plexus, and I believe I asked you all earlier in the semester if you talked about splanchnics, splanchnic nerves from 201, but essentially you can just picture these as nerve bundles that came off from important nerves somewhere near the diaphragm, and then they kind of just distributed themselves. The celiac plexus is something you can actually see with the naked eye. It's a pretty big, thick nerve bundle. But anyway, all we need to understand is that they're sympathetics, and the sympathetics are going to be important for changing the rate of how much the blood is actually coming into the kidney. So changing blood flow. And it'll also stimulate the release of renin as well. So sympathetics will change blood flow by increasing it if sympathetics are on, or decreasing it if sympathetics are off. And then we have the release of renin as well, which is also driven by sympathetics, which makes sense because sympathetics tend to increase cardiac output. So it makes sense that sympathetics would also do that by stimulating the release of renin. Okay, so we have our nephron, so this tube that I've just kind of arbitrarily drawn as sticks, and we need to identify some structures in it. So now on your next sheet of paper, let's draw our next big structure. All right, sorry, I just want to make sure that we do this in the most efficient way possible, so we'll stay with orange here. So let's draw kind of a bubble, and that bubble turns into a tube, and then that tube turns into a long tube. Oh, wow, good line. And then a short part of the tube, and then another part of the tube, and this part of the tube, and then this part of the tube. Okay, so each of these parts are going to have different names. And so let's go ahead and name them one by one. So the nephron is the entire structure, the nephron. That's what you can title your entire page if you'd like. From there, let's identify the renal corpuscle and the renal tubule. So the renal tubule is going to be everything that's tubular. Oh, and sorry, I drew the line a little far too over. So including everything to here, this is renal tubule. Okay, everything to the left, that circular piece, is going to be the renal corpuscle. Okay, and each renal tubule will dump into something called the collecting system. So this is just one branch of the collecting system, but it'll all eventually dump into. So this is actually the collecting duct, which is just one part of the collecting system, just one tube out of the collecting system. Okay, so the renal corpuscle, if we were to zoom in to the renal corpuscle and identify some structures, we could identify, and actually let's start this on our third page. Okay, so now let's zoom into what that ball looks like. So I'm going to draw like this, and like this, and like this, and like this. I would recommend drawing it as big as you can, maybe a third of the size of your paper, maybe even half. Okay, so this is our renal corpuscle, and within the renal corpuscle, we have something called we have our glomerulus. Let's draw that. All right, and remember our glomerulus got blood in from the afferent arteriole. And actually, I'm sorry, let's draw that as a straight tube. So this is our afferent arteriole, okay? And then blood leaving is going to be leaving via the efferent arteriole. There's a reason why I'm drawing it like a tube. That'll come later. Okay, all right. So now we have this space between the glomerulus and the renal corpuscle. And so the space is going to be called the capsular space. Okay, also known as Bowman's space. All right, so all these names used to be after Bowman. So if you're researching the interwebs and you see Bowman, that is still, it's not anatomically correct anymore. We're trying to phase out that name, but the diagrams are probably correct. Let's see. And then if we just talk about the circular part of this section, so not including the efferent and afferent arterioles and not including the part of the tube that's coming out of it, we would call this whole thing the glomerular capsule, or Bowman's capsule. Okay, but what? That sounds a lot like corpuscle. What's the difference? So the capsule is going to include only the glomerulus and the space that the glomerulus takes up. The renal corpuscle is going to include everything that comes in and out of it, and that's really it. So the corpuscle includes everything. The capsule is specific to identifying the actual outside boundary of it. Capsular space is within that. So renal corpuscle, big umbrella term, sub-anatomical terms from that would be the ones that we've identified so far. Okay, so we'll come back to both of these pictures, so don't lose those quite yet. Here we have a description of what's going on in the tubes, in the tubular part. So first we start with our renal corpuscle, and these are all going to be made out of squamous type cells. This makes sense because we have a lot of filtration happening here, well, we don't have any filtration happening with the glomerular capsule. We have filtration happening with the glomerulus, but we just need something to hold this space together. So it makes sense that we have some squamous cells there. Then in the first part of the tube, which I guess we should name that next. So let's go back to our renal tubule here, and now we can name our pieces. So we had our renal capsule on the left. The first piece coming off of that is going to be the PCT, and the PCT stands for proximal convoluted. Let me make sure I spelled that correctly, and I didn't. Convoluted tubule. All right. From there, we have the nephron loop. And the nephron loop consists of two parts, one coming down and one coming up. The part coming down is going to be called the descending limb of the nephron loop. The other part is coming up, and this is going to be called the ascending limb of the nephron loop. And then for every proximal, there's probably going to be a distal, so this is going to be our DCT for distal convoluted tubule. And then from there we dump into the collecting duct, which we already identified. Okay, so now that we have those labeled, now if you wanted to add in here what type of the cell types were, that's fine. I just wouldn't do that quite yet. I would wait for us to finish everything that's going to go on in this picture before you do that and see if you have enough room. You're more than welcome to after we're done, but that would just be my recommendation. Okay, so we have the squamous cells. Then we have, in the proximal convoluted tubule, we're going to have cuboidal cells with lots of microvilli. So if we have lots of microvilli, that probably means they're going to have something to do with absorption, which is definitely true. And we have cuboidal cells, which means it's probably going to have something to do with secretion. That's also definitely true. Then in the nephron loop, we have two different parts. We identify in this class them as descending and ascending, but the descending limb actually has two parts. It has a thick descending and then a thin descending, and then the ascending limb has two parts also, a thin ascending and a thick, first thin and then a thick ascending. We just simplify both of those sides into descending and ascending, but there are multiple parts to both. So descending limb will have both squamous cells, or will have mainly squamous cells, and then the ascending limb will have squamous cells and then eventually cuboidal cells. So squamous cells means maybe not necessarily absorption, but we probably might have something diffusing across those. That'll be important. And then the cuboidal cells mean we have something to do with secretion, which is definitely true, and that'll be important. Then in the distal convoluted tubule, again, we have cuboidal cells, but we don't have a lot of microvilli here, which means we probably aren't doing a lot of absorption, but maybe we're doing some secretion. And then finally in the collecting duct, part of the collecting system, again, we have a lot of, not cuboidal cells, sorry, but columnar cells. And in between those, we'll have things called intercalated and principal cells. In this class, we don't go through the specifics of the difference between those two. Essentially, they are reabsorbing water, and those two cells have specific co-transporters that we don't specify in this class, and that's why they're differentiated like that. So just focus on calling them columnar cells, and that should be sufficient. All right, so now back to this picture. Let's go ahead and label everything that's in this slide on this picture for completeness. Okay, all right. So the glomerulus is focused on collecting or filtering out our filtrate and to make our preurine. So when blood comes in through the afferent arteriole, it circulates through the glomerulus. Some stuff stays in and exits via the efferent arteriole, and some stuff comes out. So the stuff that comes out we actually call filtrate. And filtrate is going to go down through the proximal convoluted tubule and out through the rest. How we do that is through a couple of mechanisms, but the primary way the glomerulus can do this are with structures called podocytes. So I'm just going to draw little dots sporadically around here. And these are going to be podocytes. So the prefix podo means foot, cytes means cells, and these microscopically literally look like tiny feet, more like chicken feet, grabbing onto the glomerulus. These are important because they're relatively evenly spaced, but they are spaced apart from each other. So even though I only drew a couple here, they're actually more like this along the glomerulus, allowing space in between each of them. So the podocytes can actually contract or relax and allow more or less things to come in and out or out from those blood vessels. In the picture on the left in your lecture notes, the podocytes are shown here on the right side, but I think this view is kind of confusing. So what it's trying to show you here is the glomerulus, one part, or the white space at the top and the white space in the bottom are both parts of the glomerulus. It's just showing how it's folding in on itself, so you're just looking at like two parts of the tubes that might be right next to each other. And it's showing you like a red blood cell on one of them because these are really, really small, so we only are allowing like a red blood cell sized stuff to go through it, only one red blood cell at a time. And then the podocytes are these purple things that are surrounding it with a nucleus. And again, this doesn't show you like a really nice view, but if you look on the left side of this picture, oh goodness. If you look on the left side of this picture, you can actually see some of the spaces made by those podocytes, and that's where that filtrate is actually going to come out from. Okay. Okay. Next, don't go back to this picture, but just look at this picture here with me. So we draw it two-dimensionally like this. We draw it relatively flat. So with the glomerulus and the capsule on the left and the distal convoluted tubule and the collecting duct on the right. In reality, this isn't actually how it's shaped in the nephron. Like I said, everything is folded in on itself. So if you can picture the collecting duct and the distal convoluted tubule folding in on top of the renal corpuscle, then that's actually what's going on. And so in this picture here on the left in the slide, you can actually see the distal convoluted tubule is right next to those arterioles. So let's go ahead and draw that in. I'm going to draw it in orange to stay consistent because it is still part of the tubule. And I'm just going to draw a nice, I'm just going to draw a nice simple circle here. And it's a tube. We're going to have some cells, cuboidal cells, and I'm going to label this as the DCT. Okay, that's kind of weird. What's the point of doing that? So in the distal convoluted tubule, we have a specialized set of cells, let's do green, called the macula densa. And I'm going to kind of arbitrarily put it here. It's part of the tube of the DCT, macula densa. And the macula densa is actually sensing the amount of sodium that is still left in the filtrate by the time we get down there. Why is sodium important? Well, sodium likes to carry water with it, and with a lot of water comes high blood pressure, or high blood volume, high blood pressure, or high venous return, high blood pressure. So the macula densa is always constantly sensing what's actually happening to the filtrate that is still being that is still coming through the tubule. And it makes sense that the macula densa would be at the DCT, because from this picture, that would mean that it's towards the end of our filtrate. Like, we've already filtered it a couple of times by the time we got to this point, so it should be pretty much urine at this point. And so we want to be able to give some feedback back to the glomerulus and tell it, like, hey, you're doing a really good job, or hey, you need to speed things up, you're not doing too well. Okay. Then we have a set of cells right next to that. Let's go in light blue here. I'm just going to draw them arbitrarily here, like in a circle. And these are extra, meaning outside of, the glomerular cells. And in your image, they're called extraglomerular mesangial cells. It's fine, you don't have to add mesangial if you don't want to, but that would be the full name. These are going to be sensing blood flow. How good or how many red blood cells are actually coming in and out at this area. And then last but not least, the afferent arteriole also has some specialized cells that we have to account for. I'm going to draw them in like this, like little boxes on both sides. And these are going to be called our juxtaglomerular cells. Okay, well, that sounds familiar. What's that about? So these are the ones that are actually releasing renin. This is what you put in your hormone tables, probably a little bit blindly, but these are the cells that are actually going to be releasing renin. This makes sense because however much blood is coming into the afferent arteriole will brush against the juxtaglomerular cells, and the juxtaglomerular cells are going to be like, oh my gosh, we do or we don't have enough blood coming in. It's not coming in fast enough, it's coming in too slow. We need to increase the blood pressure, so we're going to release renin, and renin will activate the RAS system to eventually promote an increase in blood pressure. Okay, so that's pretty much it for small structures. We just need to add one more term. Let's go in this color. So everything from the DCT, the afferent, the efferent arteriole, all the way up to the beginning of the glomerulus. We're going to call this the juxtaglomerular complex. All right, so juxta meaning next to, glomerular meaning next to the glomerulus, and it's a complex because it's involving a lot of structures. The juxtaglomerular complex is really important because, like we mentioned with the macula densa and the extraglomerular cells and the juxtaglomerular cells, these are really what's regulating blood pressure. So the juxtaglomerular complex is the term we need to know. Okay, there's other stuff in the picture on your lecture notes. These might be helpful for you as we continue with the lecture, but for now I just want us to get these images down. Okay, so here I have a written explanation of what's going on. We've already talked about the glomerular capsule and what the glomerulus is doing, but it's just laid out for you in written terms. And now let's talk a little bit about, let's come back to the nephron picture. And here let's talk about what each part is doing. So when blood leaves the glomerulus and filtrate, enters into first the proximal convoluted tubule, it will continue to be filtered, and once it's finally filtered, then it becomes tubular fluid. So I'm going to probably call it filtrate, pre-urine, tubular fluid. Those are all going to mean the same thing, but filtrate is pretty specific to just the area of the glomerulus. So you're welcome to call it whatever you'd like, one of those three terms as it goes through the tube, but at the glomerulus, please keep it filtrate. Okay, so then tubular fluid enters after the distal convoluted tubule, then it goes through the collecting duct, and from the collecting duct it enters the papillary duct, and then a minor calyx and a major calyx, and the renal pelvis, and the ureter, the bladder, the urethra, and out. All right, why did I bother spending this much time drawing it out like this? Well, let's go ahead and take a look at the anatomy of it. Let's go to here, and we're going to look at just the lobe of a kidney. So here we're looking at the cortex, and down here is going to be the medulla. I'm sorry, not the medulla, but the part of the cortex that brushes up against the medulla are called the renal columns. You'll see that term in your lab practical list. And then we have the medulla, and the medulla, as you can see, has parts of the nephron. The nephron here is identified by this kind of white snail-looking thing. So here we start with the glomerular capsule, proximal convoluted tubule, nephron loop, DCT, collecting duct. Okay, I'm going to zoom out though. And this image also does a good job of showing you the blood supply. So we talked about those interlobar arteries, then those turn into the arcuate arteries, and then those give off branches. Here, this shows you the peritubular capillary network really nicely, surrounding the tube. So the peritubular capillary network highlighted in pink. And then we also have some extensions of that coming down into the nephron loop. We'll talk about that a little bit later. But I like this visual because it does a good job of showing you what happens to the pre-urine as it gets into the collecting duct. So once it reaches the collecting duct, it's going to dump with all the other collecting ducts into a space called the renal papilla. And from the renal papilla, then it enters into the minor calyx. So renal papilla was not on the previous model that I showed you because it's a little too small and the angle is a little weird, but from this angle, it's a nice angle. In the models that we have in the classroom and that we'll post pictures of, this might be a really difficult structure to visually see. But once you've seen it in one good model, then you can see it in the other models. So I would recommend becoming familiar with the renal papilla here before applying it to the other structures. And then that fat that we were talking about before, the perirenal fat, that's actually located in between each of these minor calyxes. Okay, so this is a pretty cool model and nice and shiny. Just showing you one piece of the pie. The left side of the kidney tubule has it cut away so that you can see that it's an actual tube, and on the right side it's the same model, just enclosed. This is a juxtamedullary nephron versus this one up here is a cortical nephron, to show you the length difference between the two. Oop. Okay, I believe that's all that I want to show you on this model. So let's go back to our notes. Oh, right. We already talked about, so we talked about the collecting duct, then we reached the papillary duct, so we can draw that in here. That was not labeled on that picture. So I'll go in purple here. So here's the papillary duct. It's just a changing of the names from collecting duct into papillary duct. And then from papillary duct, we enter into the renal papilla, then minor calyx, major, renal pelvis, and so on. And we already mentioned those earlier. Okay, all right. Let's see here. So we already talked about the juxtaglomerular complex. We already talked about the macula densa and having chemoreceptors and or baroreceptors. So chemoreceptors paying attention to sodium levels, baroreceptors responding to level of stretch. The juxtaglomerular cells are the ones that are secreting renin. These are in the afferent arteriole. And then the extraglomerular mesangial cells, those are the ones that are, we don't specify in this class where EPO is coming from, but EPO is coming from this area, and it's providing feedback between macula densa and the juxtaglomerular cells. So helping them make sure they're giving the same signal. It would be very confusing if the macula densa was trying to decrease blood pressure, but the juxtaglomerular cells are trying to increase pressure. That seems pretty inefficient. So the extraglomerular mesangial cells are going to help us do that. Okay, in the collecting system, we have, like I said, the cuboidal cells with microvilli, and the cuboidal cells without microvilli. And like I mentioned, we don't really go into the nitty-gritty differences between the intercalated cells and the principal cells and which ones are doing what, but it's important to understand that these are cuboidal cells. The collecting system will be really important for reabsorbing of water. This table in your book goes through and gives you some pictures of what the differences are. Hi, Nikita. Gives you some visual pictures of what's going on in the collecting system, but it's more important to reference what our slides actually look like. Okay, we already talked about how our cortical nephrons are the majority of our nephrons, with the juxtaglomerular nephrons being the other 15%. Now we need to add the last bit of information to our picture here. Okay, so we have our peritubular capillary, peritubular capillaries, and that's what I've denoted in purple. Now we need to add in the vasa recta, and the vasa recta is only going to be in the juxtamedullary nephrons. And the reason for this is because the vasa recta has a really specific job that we'll talk about towards the end of this chapter. So we have to be able to differentiate between those two. So here would be an example of a cortical nephron with the peritubular capillary network versus, and I'm kind of running out of room here, but you could draw a nice bigger version. Actually, so I would draw it in this picture. I don't want to ruin what we have here. I'm just going to continue down here and make a new version. Okay, and let's call this a juxtaglomerular nephron. All right, so the afferent arteriole goes into the glomerulus, then we got the glomerulus, then we've got the efferent arteriole, and from there, we immediately go into what's called the vasa recta. Vasa recta is here. And then the vasa recta will join up with the rest of the blood supply. All right. Okay, here's a cartoonistic depiction of what's going on. So figure A shows you both the cortical and the juxtamedullary nephrons and where they are in relation to the renal medulla and renal cortex. And then B and C show you the difference between the cortical nephron and the juxtamedullary nephron. All right, see you for the next